Our next talk is by Gustavo de Miranda. It's called The Microendemics of Madagascar, Diversity and Systematics of Madagascan Schizomida. Sorry. Gustavo, please take it away.
Okay. Can you see my screen?
Yes, looks great.
Okay, thank you. Thank you for the introduction. So yeah, my name is Gustavo de Miranda. I'm currently a postdoc here at the Smithsonian, and I'll be talking about Madagascan schizomids. And here we can already see how amazing these animals are. Here's a Bucinozomus, a picture from Nick Day from Singapore that has this projected abdomen here that is telescoped, which is crazy. And here's Hubardia from California, a genus that is endemic to that region of California. But moving on to the talk, so I thought I would start explaining a little bit of who short-tailed whipscorpions are, or schizomids are. Just I know most people know, but you never know. So it's a heterogeneous group here. So schizomids are these small arachnids who got their popular name from resembling whipscorpions, which are here. And but differently, they have a short tail. So you can see here the flagellum that is very small in schizomids and is very large and multi-articulated in telephonids or vinagrums. They're part of this group called Pedipalpi, which is sister to Araneae. So they are a sister group to spiders. But differently from spiders, they are very little diverse. So the name of the order is Schizomida, and there are only 273 species. And despite being small, they are very widely distributed. So we can find schizomids all over the world, especially in the tropics, but also in the US, and introduced species to some other countries in Europe. And so yeah, the distribution is quite large. And recently there has been an increase in the understanding of the systematics of the group. So lots of new species, new genera, and also new phylogenies. So in the last few years, multiple new genera and species have been described. And here's to illustrate some phylogenies published recently from Rodrigo Monjaras Redas on Myazomus, Olmeca, Protoschizomida. And these papers that Rodrigo is publishing, and also in Australia, Mark Harvey has been working on, is mostly, so the work's mostly focused on this country's fauna, so Mexico and Australia. However, in 2017, Klaus et al. Have published that was the first large phylogeny within the group. It included telephonids and schizomids, and included taxa from all continents, and provided a great overview of the evolution and biogeography of the group. However, there was very few representatives of African or Afrotropical schizomids in the phylogeny. So they included a few species from West Africa, or specimens from West Africa, and that's it. But it was a great contribution, but lacked from the diversity of schizomids that we have from the continent. So the Afrotropics is one of the least known areas in the diversity of schizomids in the world, and not only schizomids, but multiple other groups. And right now we know 15 genera from there, from the countries marked here. And in Klaus's phylogeny, he only included a few specimens from this area. And within Madagascar, particularly, only 3 species are known right now, and they are all in the genus Bamazomus. They are Bamazomus madagassus, Vadoni, and Miloti. And here we are looking at the flagellum of these species, and the male flagellum of schizomids. They are really important taxonomic structure or appendage. And with the shape of the flagellum, projections, grooves, and position of the setae, we can tell apart, diagnose species, but not only the male, also the female. We use the flagellum of the female pedipalp, and also the spermatheca, which is a really important structure to diagnose species in general. So the currently small diversity in Schizomida in Madagascar is likely an artifact of little collecting, because the island is huge, and also we know that Madagascar is one of the world hotspots of biodiversity. There is a high degree of endemism. For example, 100% of the native Malagasy amphibian and terrestrial mammal species, 92% of reptiles, 74% of butterflies are endemic to Madagascar, occur nowhere else. Schizomids are known to be short-range endemics, as seen in Australia, and they can also inhabit a wide range of altitudes. For example, the Andes and Madagascar, and it would be expected then that the diversity of schizomyids in Madagascar is higher than what we see today. So with this in mind, this work has the aim of tackling one piece of the apotropical puzzle of biodiversity, investigating the fauna of Madagascan schizomids. Specifically, we wanted to build a phylogeny of the Madagascan schizomida to test the morphological hypothesis with the molecular phylogeny and to place the Madagascan schizomida in the order's tree of life using Claus's phylogeny as reference. So for that, we used museum specimens preserved in the California Academy of Sciences and the Natural History Museum of Denmark, as well as some fresh material that we collected in California, the Hubardia that I showed in the beginning, to use as outgroup. So here's a picture of the California Academy of Science collection, and we had specimens with age ranging from 1995 to 2018. We used arachnid UC probes to sequence loci, so we sequenced 635 loci. And from there, we fished the usual suspects, legacy loci that are used in phylogeny, such as 18S, 28S, and CO1, to place the Madagascan schizomids within the phylogeny published by Claus et al. That used the usual suspects. So here is Hannah Wood and Rowan French, an amazing undergraduate student we had here at the Smithsonian working on this project. This is us collecting and preserving arachnids in general, and also schizomids in liquid nitrogen for the Smithsonian Biorepository. So, okay, so for fishing out the sequences, the genes from the UC sequences, we use the Phylogenetics pipeline. And I'm not going into much more details in the methods, but I'm happy to discuss anything else that anyone wants to know further. So as a result, so here's an overview of the morphological diversity of schizomida from Madagascar. So here in the left, we have pictures of the spermatheca, and here on the right of this image, we have the male flagellum. So we categorize the specimens that we had in 9 different genera, and here they are. Not all of them had males and females, and it was hard sometimes to associate when they were not from the same locality. But some of them we had both the male morphology, the male flagellum morphology, and the spermatheca, and we were confident in telling them apart. Others we had only the spermatheca or the flagellum, but that's what we got. And so the morphology and diversity was way greater than previously known and thought, and followed the expectations for such a diverse region of the world. And so here's the distribution of what we found. So the yellow squares are the 3 previous records of the 3 different species that we knew from there. And the yellow dots are the new records that we are putting on the map with this work based all on museum material. And so, yeah, so there is a significant increase in both in the morphological knowledge on the group and also in the distribution in the island. So then we wanted to understand the evolution, the phylogeny of the group. So we took the sequences that we sequenced. We run a maximum likelihood analysis in iQuid3, using 150 loci in a matrix of a 50% completeness. And not all specimens that we put in analysis that we sequenced had useful sequences for analysis. So we got, for example, genus 1, 2, and 6 and 4 recovered as monophyletic. And we also included Bamazomus, which is the genus that is known to occur from Madagascar. But it also occurs, I forgot to mention that Bamazomus also occurs in Vanuatu and Australia. And of course, this is important because we wanted to understand the biogeography of the group there. And we recovered Bamazomus as a monophyletic here, and the other groups separated from Bamazomus. And these other specimens are specimens that we did not assign to any genus or species because they were mostly juveniles, and they didn't have developed genitalia for naming them. So here's a plot of the phylogeny, the map, for us to understand where they are from. Okay, so for a better idea of the distribution, here is a plot of the map in the country, And interestingly, the older taxa are on the eastern part of the island, and the newer taxa, the more recent taxa, are here on the top and western part. And this might be interesting biogeographically. I'll talk a little bit about this in the next slides. So yeah, so this is what we got for, within the schizomids in Madagascar. So as I mentioned, we fished out the usual suspect genes from the UCs, and we downloaded sequences from GeneBank, mostly from these two works, from Klaus et al. And from Rodrigo Morjaras Redes et al. In 2019. And then we ran analysis with all the data together. And what we found was that the Madagascar schizomida is sister to the Oceanian species and genera. And it is also closely related to Australian groups.
Gustavo.
Okay, thank you. But not Bamazomos, as previously thought. So here with these arrows, we can see some taxa from Australia. So it's a new genus that Klaus et al. Proposed from Australia. Bamazomos is here, Draculoides is here, and the specimens and species from Madagascar are here, more closely related to other Indo-Pacific and Oceanic species, not from Australian species. And this remounts to either a Gondwanan distribution of the group or recent colonization. So the Madagascar fauna, they are composed of both things, of Gondwanan, relict Gondwanan species and recent colonizations, and this could be the case for some of the taxa of the Schizomida in Madagascar. So our findings corroborate what Klaus and collaborators demonstrated with specimens from America colonizing West Africa, while the Madagascan Schizomids derived from Australian species and from oceanic species, especially from this area here. And this work is going to be the scaffold for further biogeographic analysis that we haven't run yet, but we are going to run very soon. And with that, we found a way higher diversity than previously thought in the island. We also interestingly found that the morphological classification was consistent and found consistent in the phylogeny. This was the first time high throughput and Sanger sequences were combined for the group and showed the potential for using the legacy data that is in GeneBank, for example. And it also shows that an ancient orange and diversification within the island for schizomids. And with that, I would like to thank the institutions that were involved and donated material for this study, and also colleagues that helped with molecular work, and also Mark Harvey and Kim Abrams, who donated sequences, extractions of the Australian Bamazomos. And I would like to thank you for your attention, and I'm open for questions.
Thank you, Gustavo. Very good talk.
Thank
you. So we have some questions rolling in. So from Ivan, he asks, how successful is fishing Sanger markers from UCEs?
That was the most challenging thing. It's not that it's that hard. The problems like making sure that what you fished out was the sequence itself and not contamination or some crazy random sequence. So I had to blast all sequences to make sure they were schizomids, and they were like what we expected. And the problem is that we don't have the same sequence. Not always we are able to fish out the same gene for all specimens. So sometimes I have one gene for these specimens and a different gene for another specimens, and it's hard for them to, when I run analysis, it's hard for them to, for example, I know it's a gene 1 species 1, but I got different genes for them for whatever reason, because that's what I got from the UCs, and then I don't get them in the same clade. So one option can be kind of to create a chimera and combine different ones if we are sure they are the same. But fishing out, it's not that hard. It's hard then afterwards to make sense of everything, because it can be tricky sometimes.
Mm. Another question from Rodrigo. Any luck collecting Megaschizomus? And he has a follow-up question. And which probe set did you use?
I used the arachnid probe set, not the spider probe set. And it was fine, like 150 genes was not bad. We only had like Sanger sequencing before. And mega schizomes, I never seen one fresh in collections to including, or fresh enough at least to including in the analysis, because you have to remember the schizomids are tiny, and we grabbed like 2 or 3 legs to sequence. So really old specimens are very hard to sequence. That's why we went up to 1995. So yeah, we would probably need to have more recent ones from collection, and I haven't seen any or know of someone that collected.
We have another question from Prashant. He says,
Yeah.
wonderful talk. One of the challenges we faced in the Klaus et al. Paper was rooting the schizomida tree. How did you select your outgroups, and have you been able to sequence proto-schizomids?
No, I haven't been able to sequence proto-schizomids, and I confess I had also like trouble rooting the tree to get sort of the same topology. But in the end, what I did, so I selected all samples and I got all. I took their spreadsheet where they said which specimens were used for the big phylogeny that they published there, and then I filtered to have few representatives of each group, because I didn't need all that they used there. I needed only a few representatives of the specific taxa, and then it worked out. I chose the outgroup based on their phylogeny. So Hubardia was recovered as the sister to all Hubardiidae. So I used that for our analysis within Madagascar. So that was our choice based on their paper, based on their results. But
Okay.
It can be tricky.
Yeah. One last question from Hadeen. He says, great talk. And he wants to know what is happening in the Seychelles.
That's an amazing question. There's so many genera and species in the Seychelles. And I don't know what's happening in the Seychelles. I want to know the answer too. It's really impressive. I didn't have any to include here, but it's crazy because it's a small island with a huge diversity. Wow. It's a very good question.
Well, thank you very much.
Thank you. Thanks everyone.
Our next